The Parkinson's Podcast is brought to you by the Davis-Phinney Foundation and brings you the stories, wisdom, and expertise of people living with Parkinson's, care partners, and Parkinson's professionals. In this episode, Chris Kruger, Davis-Phinney Foundation Senior Content Manager, talks with Kevin Kwok and Dr. Pinky Agarwal about gene therapy for Parkinson's. Kevin has lived with Parkinson's for over 15 years and has professional training as a pharmacist, which he brought to his more than 25 years as an executive in the biopharmaceutical industry. Today, Kevin participates on multiple clinical trial advisory boards and is an active volunteer and advocate. Dr. Agarwal is a movement disorder specialist currently practicing at Evergreen Health in Washington. Dr. Agarwal is actively involved in clinical research, including as an investigator in the Regenerate PD gene therapy trial. This episode has some terminology that may be unfamiliar. For convenience, we include a list of definitions for some of these terms in the description of the episode. This episode is sponsored by Ask Bio.
Kevin, Dr. Agarwal, thanks for joining me to talk about gene therapy in Parkinson's. To start, maybe we'll go at a high level here, and I'll ask first, Dr. Agarwal, what are genes? How are they involved in Parkinson's?
Genes are stretches of DNA that provide instructions for making proteins, which in turn control how our cells function. Now, in Parkinson's disease, certain genetic changes can disrupt normal processes within the cell, which then leads to some protein buildup within the cell. So while most cases of Parkinson's are not inherited, approximately 10 to 15% can be linked to genetic causes. And there are mutations in genes such as the SNCA, the LOCK2, POC2, PINK1, and GBA, which may increase the risk or directly cause familial forms of Parkinson's disease. And these discoveries highlight that both inherited variants as well as subtle genetic risk factors contribute to disease onset and progression.
Okay, thanks. Thanks for that answer. So gene therapy, what is that? How does gene therapy work and how does that relate to Parkinson's? What's its status in treating Parkinson's? Are there any gene therapies now? Can you just talk about that from a high level? What's gene therapy? How is it used in Parkinson's today?
Sure. So gene therapy refers to a series of strategies that either modify the expression of a gene or correct a gene that is dysfunctional. And the aim is to treat the root cause of the disease itself. So gene therapy involves a series of strategies that will modify the gene expression or correct the faulty gene. And in Parkinson's, there is currently clinical research exploring approaches such as restoring dopamine production, because we know dopamine is decreased in Parkinson's, protecting nerve cells from further damage, because we know over time the dopaminergic and other nerve cells die or decrease production, and correcting underlying genetic dysfunction. And this is being studied in several clinical trials currently underway.
Okay, so it's in trials, but there's no currently approved gene therapies for Parkinson's today, right?
No currently approved gene therapy, but trials underway.
As far as I know, not all gene therapy trials today are seeking to treat people only who have a genetic variant that is known to be associated with Parkinson's. There are genetic therapy or gene therapy approaches that treat people who don't have necessarily have a mutation or a variant. Is that true?
Exactly. So, gene therapy, there are some gene therapies that are targeted to people with specific mutations, such as people with GBA or LOCK2. However, there are several gene therapy strategies that are looking at shared disease mechanisms that are common for all people with Parkinson's disease. So we know that in everyone, all Parkinson's patients, irrespective of genetic status, there's dopamine loss. There can be circuit imbalance. There can be nerve cells that are vulnerable to further damage. So there's more than just single genetic defects. And so a lot of these gene therapy trials or mechanisms that are currently underway may benefit the broader Parkinson's population, not just people who have specific genetic mutations.
Okay, great. Thanks for that broader context. I appreciate that. And Kevin, speaking of broader context, what's your perspective on this topic so far as a person living with Parkinson's?
Well, I'll tell you, I'm really excited about the way there's been an influx of different modalities being explored for the treatment of our disease. And leading that excitement is, in fact, gene therapy. For me, this is getting at the root cause of disease and not symptomatic symptomatic sort of putting out the fire, right? This really gets at the essence of why we have, from a biological standpoint, the disease that we live with. With that, however, I have concerns and cautions on safety.
Yeah, Kevin, I hear people ask questions about the safety and raise concerns about the safety of gene therapy from time to time. And sometimes it's about the surgical procedure that's often involved. But another thing that people seem to be concerned about is the virus that's used. I guess it's usually an AAV virus, I believe. I think this causes people concern. And I think maybe sometimes they're concerned that they might be made sick by the virus. But Dr. Agarwal, can you comment on this? How are viruses used in gene therapy?
So, what they're using is they're using the virus basically only as a delivery mechanism because you have to get something into the brain. And so the AAV vector, it's just being used to carry a genetic payload. And that's not integrating into the patient's chromosome. It includes mainly three parts. There's a promoter part, and there's actually the therapeutic DNA. And then there's another part that's a stop signal. So basically what happens is that the promoter part, it's a regulatory DNA switch that tells the machinery when, where, and how strongly to make the protein from the introduced gene.
So when you say machinery, you're talking about the machinery inside of a cell that is actually what produces whatever we're trying with the gene therapy to make happen.
Exactly. And then there's the transgene, which is the actual therapeutic gene that's placed inside this viral vector. For example, that transgene can be a transgene for CDNF, for AADC, for GCH1, and that is encoding for the protein that we need to improve the cell function. And then finally, there's a stop gene that'll tell it to stop making more when it doesn't need to make more.
Okay, so this is a process where the virus is going to deliver three pieces to the cell, and those pieces say, "Hey, you're gonna start doing something. Here's what you're gonna start doing, and oh yeah, don't forget you got to stop at some point."
Exactly.
And that is not necessarily changing the way the DNA in the cell. It's just addressing the pieces of the cell that make things happen. Not going into the DNA and saying change DNA, be different than you are. It's saying machinery, act differently right now.
Exactly.
So, Dr. Agarwal, that's maybe a bit about how gene therapy is delivered. But I wonder if maybe you could say a little more about what gene therapy might be able to do to help in Parkinson's. What are researchers trying to use gene therapy to accomplish with the viral vectors you just described?
So the main strategies that are being used for gene therapy, again, is if you look at basic mechanisms that they're trying to address, one is just dopamine restoration. Now, how can we restore dopamine? Well, we know that the AADC, it's the enzyme that enhances conversion of levodopa to dopamine. So there's some gene therapy that's designed to increase AADC. Then there are some that try to boost dopamine synthesis by increasing tyrosine hydrolase. Then there is GCH1 that produces a cofactor that's critical to dopamine synthesis. Now, these don't necessarily have to be used in isolation. So they're looking at strategies to combine all of these because any mechanism by which we can enhance dopamine production would be useful. Then there are other sets that are looking at neuroprotection or growth factors. And so those growth factors, there's GDNF, which promotes the survival and the functioning of the dopaminergic nerve cells. Then there is the nurturin that was used for, a family that supports dopamine nerve cells. Then there are several other growth factors such as CDNF. And then there is the mesencephalic astrocyte neurotrophic factor. So these are aimed at protecting the remaining neurons slow neurodegeneration, and hopefully improve long-term function. But there's also mechanisms such as circuit modulation, which can rebalance the abnormal signaling in Parkinson's. So one of the main ones being looked at is GAD or GAD, which encodes the enzyme that converts glutamate into GABA, which is an inhibitory neurotransmitter. So the idea is to deliver the GAD to the subthalamic nucleus, sort of similar, how we try to influence the subthalamic nucleus with DBS. We try to change the circuitry there. So delivering GAD to the subthalamic nucleus reduces overactivity and helping to rebalance the motor circuits.
Okay, so let me try to run back what you just said so I can make sure that I understand it and maybe help our listeners too, because you referred to a lot of different substances that may have been affected or maybe impacted by a gene therapy approach. But I think you said three main categories of techniques or things that are attempted. One is to support dopamine synthesis or production of dopamine by working on a couple different biochemical products in the brain. Is that right?
Yes.
So that's one way. The second way you talked about, I think, was supporting neuronal health to protect neurons by giving increasing supply of GDNF or other substances. Is that the second way?
Yes. And then the third is circuit modulation,
which is similar to what happens with DBS, where we're sort of helping the circuits that are involved in Parkinson's function more normally than they would if there was no treatment being delivered.
Exactly.
And so those are the three things you described, and I think I got it. Did I do a good job?
Perfect.
Okay, great. So Kevin, do you have any thoughts about this? I mean, there's a lot of that and I'm just wondering if you have any thoughts about the person with Parkinson's experience or perspective on these different approaches.
My question is, where is it most effective? Is it effective in late stage Parkinson's, where you have the chance of reversing Parkinson's disease, or is it in the early stages as prevention, in some ways like a therapeutic vaccine? So for me, the question is, not all these patients may react the same way. And I think that's where I'm hoping the current studies will lead us in directions to give us the right view.
And I think the answer has to be we don't know yet, but that's the thing we have to try to figure out is who does this help the most? When is it best delivered? Do you have any thoughts about that, Dr. Agarwal?
So I think currently most of the gene therapy strategies are being designed for people who already have symptoms. And we are trying to restore dopamine function, protect nerve cells, rebalance abnormal brain circuits. Now, preventative use in at-risk individual is a possibility for the future, not currently focus of active clinical trials, because again, these are being delivered. There is some risk from these procedures. And I don't think we have enough data right now to put it in at-risk individuals. Now, in the future, people that are therapies that are in for people with certain genetic mutations, it may be maybe early intervention will be for people with known genetic mutations. But for by and large, because the mechanism is aimed at restoring dopamine, we are using it for people who have known dopamine decrease or dysfunction, and that's why it's being used in people who are already diagnosed with Parkinson's.
Well, thanks for that answer, and thanks for the question, Kevin. You brought a new sense of what we were going to talk about today to my mind, and I appreciate that. So there's a lot of things that people think about gene therapy when they hear the term. They hear gene therapy and they think of, that it must mean editing genes, or it must mean changing DNA on a species-wide scale or something like that. So I wonder if we can maybe jump in and start talking about some of these, I'll call them myths. Maybe that's not fair. And if you think that's not fair, please feel free to tell me I'm approaching it wrong. But maybe you've already talked about one of these, namely in terms of Parkinson's, that gene therapy for Parkinson's is only relevant for people with genetic mutations like GBA and LRRK2. That's not true. We've already discussed that. Is there anything else to say about that we haven't already talked about?
No, because a lot of these are looking at different at mechanisms that are affected downstream, however one gets Parkinson's, whether it's of genetic etiology or environmental etiology. We know dopamine can be decreased, brain circuits can be altered, and so these gene therapies can have widespread use.
I'm just thinking about, Kevin, I know you have DBS, and I'm just thinking about the fact that maybe that's a question we don't know about yet, whether if you're taking a circuit modulation approach to gene therapy, how that would intersect with DBS. I don't know if there's any information about that or if there's anything we know about that already, but it's certainly a question for me. And I wonder if either of you have any insights or thoughts about that.
Well, I know that as I look at the studies that are currently available for people with Parkinson's, most of those studies rule out people with DBS. And to me, that's a real frustration, because if one is controlled on DBS, it would be the same as saying someone is controlled on a carbidopa-levodopa regimen. And so for me, I think that as I look at where the field goes in Parkinson's, we will eventually need a cocktail of different therapies. One could be gene therapy, the other could be traditional medication, the third could be deep brain stimulation, and the fourth may be lifestyle things like exercise. And it's the combination of all of those that lead to the personalized care that I'm interested in and believe that will help me long term. So for me, I'd love to see silos broken down, and gene therapy, people talking to DVS, people talking to therapeutic researchers, so that we can all look at a more holistic way of treating us.
So, some of the limitations nowadays are for people with DBS, right? It's because, one, you need several MRIs for accurate placement. So DBS may just impact people's ability to get those high resolution MRIs. Now, should any of these therapies get approved, especially ones that are not directly put in the brain, like if they are given intrathecally, or if they're giving intravenous route, in that case, people with DBS will likely be able to participate. It's just when it's in the brain and you already have leads there and you're trying to aim for the same place, the putamen, or, in some the subthalamic nucleus, you're looking at to put the gene and the gene therapy in exactly the same place where you already have a device in the brain.
I have another thought about this too, which is that I also share Kevin's sense of frustration, although mine comes from a slightly different place. I haven't said on this call yet, but I also live with Parkinson's, and I have I'm still relatively well controlled with just medication, and it's going okay. But when I think about my future, I think, well, I know I'm interested in participating in trials, and I think it's coming to the point where I may be approaching the point where my movement disorder specialist is going to say, maybe we need to think about DBS sometime. And I hesitate about that decision. I will hesitate about the decision because of the fact that I know the trials that I'm interested in may not admit me into the trial or enroll me in the trial if I had DBS. That's a minor concern in some sense, but it's also another layer. That's not a problem with gene therapy. That's a problem that you face no matter what trial you might be interested in, but it is a factor in my mind that I face as a person with Parkinson's.
If you'd asked me this same question 5 years ago, I would say don't wait for DBS. Today, I would say you should look at a broader array of options, including gene therapy trials like the one is just beginning.
And at the present time, if you had gene therapy, you can go ahead and have DBS in the future, but it does limit, if you've already had DBS, which potential gene therapy trials you could enroll in the future, especially, but like if it's intravenous or intrathecal, there might be flexibility.
Can I ask a question, Dr. Agarwal? My feeling My understanding in the field today is that several of these gene therapies are one-time installation surgically. And so it's sort of one and done in that situation, which could be marvelous if it worked. But if it had some form of side effect, like an overproduction of dopamine, have they actually seen anything in any of the animal models that showed that there could be an irreversible toxicity?
So far, we are not seeing any reports of excessive dyskinesias related to these therapies.
Have they seen any issues of impulse control behavior changes or as a result of stimulation of dopamine?
Very good, a very pertinent concern. But because with these, you don't have the pulsatile stimulation, and two, you're just making dopamine. So it's more like taking carbidopa, levodopa, and less like taking an agonist. And the agonist, typically we see more of the impulse control issues with the agonists. Now the agonists selectively bind more to the D2 and the T3 receptors that are more involved in these impulse control issues. Levodopa or dopamine is binding to all of the receptors D1 through D5. And that's typically less involved with impulse control issues. So to answer your question, in the trials, have they seen it? Not that I'm aware of.
So let's go to the next quote unquote myth of gene therapy. And this is, maybe I'll combine two of the questions we have on our plan here to say, here's the myth. Gene therapy means you're editing your genes and that this can be dangerous and make species-wide changes in the long run. What do you think about that, Dr. Agarwal?
So, gene therapy, the way it's being used in Parkinson's, is not altering the human DNA, and it cannot be passed on to future generations. And the reason is that the current therapies, when they're using the viral vectors, it is delivering the transgene, first of all, only into specific brain cells. And though it's inside the cell, it's outside the chromosome. And it is not integrating into the patient's DNA. And so it cannot be inherited. And so these treatments, they're tightly regulated, they're in very targeted areas, and hopefully they'll improve the cell function, but they will not rewrite the person's own DNA. Now, there are some other things that it may be confused, such as with CRISPR. CRISPR, you're trying to actually edit the patient's own gene. And that's not what's being done in gene therapy trials in Parkinson's. You're not doing anything to the patient's own DNA.
I think maybe we can transition to talking about the gene therapy trials that are currently underway for Parkinson's. So what's happening in gene therapy trials today for Parkinson's?
So, several clinical trials, some, like we discussed, are trying to replace dopamine. So trying to place in the AADC, the tyrosine hydroxylase, or the GCH1 gene delivery to boost dopamine synthesis. And then there's a circuit modulation, the GAD. And then immediately we have the GDNF trials that are going on, which is basically using the AAV virus to put in GDNF, and that trial is currently recruiting. And then there are other trials that are looking at patients with GBA mutation. And the idea is to replace the glucocerebroside activity in patients who have less of it because they have the GBA mutation. And so we discussed that there's the AAV vector, right? But then there are some newer biotech companies that are engineering that AAV viral vector into a next generation that may have improved brain penetration. So what they can do is, right now, some of the gene therapies we discussed, you have to put into the brain, into the putamen. But if you have improved brain penetration, then you can give it intravenously. And so some of these companies are looking to give the therapy intravenously rather than through brain surgery into the putamen or into the subthalamic nucleus.
So that sounds like it relates to the next question I was going to ask, but which is about the future of gene therapy trials. But maybe I want to pause for a second and see what Kevin has to say about any of those trials that he just heard about. And do you have any thoughts about that, Kevin?
No, it's just that I'm really excited as a patient that the field is opening up.
Yeah, I share your excitement, Kevin. I think there's so many interesting approaches that you were just describing, Dr. Agarwal. And, I think that the fact that there's not only are there different targets being addressed in these trials, but also different methods to execute them. That's a bad word to use when we're talking about doing things to the brain. We don't want to use the word execute probably, but to deliver the therapy. Kevin and I have said that we're excited. What excites you about these trials?
I think what really excites me is that it's moved from a distant field with real clinical trials, real tangible data, and there's growing optimism. These trials are still early, but they are trending in the right direction. I think we are getting more safety data and effectiveness. We are looking at not just clinical outcomes, but also biological outcomes like PET scans to see how much dopamine the brain's actually making. Now, any of these trials, right, if we have to have unbiased judgment, we do need to have sort of blinded studies in which both investigators as well as patients are not aware. So what we are getting is real data that's not biased by people thinking, oh, I got gene therapy and so I'm doing better, or providers thinking, oh, knowing that the patients have had gene therapy. So, I think the field is evolving very quickly and in the right direction. And I'm hoping that we'll have not just symptomatic options, but real therapies we can offer that will slow progression in the long run.
Is there any other question you have about gene therapy and Parkinson's, Kevin? Is there anything else you think we didn't talk about that we should have?
Well, I thought one thing that is always on my mind is sort of the when you create these novel new interventions. Gene therapy is something that I, in my mind, believe is a very expensive process. And the question that I have is creating a system where there's the very privileged get access to these therapies. And it's almost a two-class system if you don't qualify. I think researchers need to think about ways that if they do succeed in getting things developed, they develop a offering that all patients can benefit from. I realize that's a little naive to think, but to me, it's very important if we're really going to create equity in the medical care system.
That's a great point to bring up. And so if you look at it, Parkinson's currently, when we don't have anything that is known to slow progression, is a very expensive condition, both for patients, their families, the fact that people may have to retire earlier or go on disability, and in general to the whole cost system. Now, I don't know an exact price and, it won't be known for a while, but if we have a one-time surgical procedure that has the potential to slow progression, it could actually be a big cost savings to everyone.
Sure.
Now, there are other gene therapies that are being covered by health insurance systems. And currently, some of the gene therapies that are being paid for are, children, in which there's also a question of survival or not survival. Now, in Parkinson's, again, like I said, it's a huge cost to the whole Medicare system. So if there is robust data that it slows progression, and, they can do health economics where they're looking at people who have gene therapy, how much is being spent on their hospitalization, caregiver, all of those health economics data, compare it with people who are getting standard of care at what we have right now. And if there is a cost savings, I don't know. I mean, it would make sense for Medicare and other insurances to cover it.
Yeah, this is a really good point, Kevin. So thanks for bringing it up. I think that, as the Davis-Phinney Foundation, we're focused on living well with Parkinson's today. And that doesn't mean one group of people living with Parkinson's. That means people with Parkinson's. We want to help everyone live well today. So this is very close to the mission of the Davis-Phinney Foundation. Well, Kevin, Dr. Agarwal, I really appreciate you taking the time to talk about gene therapy with me. It's been informative to me, and I hope it's been informative to our audience. And I hope we can talk again sometime soon. Thanks for your time.
Thank you very much.
Thanks, Chris.
Thank you for joining us for this episode of the Parkinson's Podcast. For more information about the Davis-Phinney Foundation and to learn about educational offerings and community events for people affected by Parkinson's, please visit davisfinneyfoundation.org or dpf.org. This content includes information, insights, and opinions from our Parkinson's community. The views expressed are those of individual participants and do not constitute medical advice. This content is not intended to substitute for the guidance, advice, or recommendations of your personal healthcare providers.